Hi everyone. Welcome to our BioBuilder Club webinar of the day. Dr. Danielle Gutillet is back with Biodesign 102. So last time, and I'll make sure that in my follow-up email, the video to Biodesign 102 is linked. So this time you are going to hear the parts level design. And I will also be sharing the slides with teams later so that you can refer to these beautiful graphics. All right, you want to take it away, Danielle?
Sure. All right. Hello, everyone. If you were here last time, you know that we like trying to make it a little interactive as much as we can on here. So please feel free to use that chat. If you missed the last one, no worries. We're going to do a quick recap before we dive into parts. But there will definitely be parts of the webinar where I will ask for your kind of feedback or contributions. So definitely get that chat going, and which reminds me to open the chat. I should probably do that. Here we go. All right, I've got my chat open.
I'll monitor the chat too and let you know if we're ignoring.
Perfect, perfect. All right, so let's get started here. Okay, so today we're going to be talking about parts level design for our project. So a quick agenda of what we're going to be covering. If you missed the first webinar, no big deal. We're going to do a quick recap of the topics that we covered before we dive into parts level design. So we'll quickly go over what we talked about in terms of topic, chassis selection, device level design, input, output, all of that. And then we're going to revisit the arsenic biosensor example from the last webinar that we used, and we're going to use that system to walk through how to build genetic circuits and then how to find parts for your genetic circuits. Okay, so last webinar, we talked about the biodesign abstraction hierarchy, and your BioBuilder Club handbook walks you through the steps of this whole process and how to use it to design your project. So hopefully you have been using that resource because that is a fantastic resource for you. Last time we discussed different ways to zero in on your topic area. We talked about identifying a challenge area that you'd like to address. We talked about brainstorming different approaches to solving that challenge and how there is no one solution to every problem. So keep that list going of all of your different ideas. Then we talked about how to decide on an approach. So again, we encourage you to come up with multiple topic areas because as you begin to research and design your project, you might want to end up switching to a different topic. One might be like a dead end, or it's just not working out, or it's too complicated. So it's nice to have another idea already kind of fleshed out to fall back onto. So from there, then we started to talk about system level design. So for system level design, a lot of decisions are being made at this stage, but you can be super vague still. So think of system level design as just like a one sentence description of what you want to achieve with your system, just super basic level. So it could be a sentence like, we want to engineer blank to do or produce blank in response to blank. Just that simple. And those blanks could be very vague. It could be, we want to engineer bacteria to break down plastic in response to whatever, seeing plastic, right? Or sensing plastic. So you want to define your overall purpose or function for your system. And the three most common purposes of synthetic design are listed here. So a system that functions to produce a specific protein. An example here would be biofuels or any sort of pharmacological product, right? Drug production. You just want to design a system that pumps out a lot of this protein product. Number two is a biosensor. That would be a system where you want to design a system that senses the presence or absence of something and indicates that to you. That's the one that we're doing our example on, the arsenic biodetector. And the third most common is transforming one chemical compound into another. Okay, so it's likely that your group has selected a system that does one of these three things, which is great. So once you decide on the purpose of your system and the general functionality, like how you want it to work, the input and the output, Then you're going to choose your chassis. Remember, chassis is just our fancy word of whatever our host is. It could be a bacteria, it could be yeast, or we talked last time about using cell-free systems. So if you want more information on that, you can go back and revisit our first webinar. Then we discussed the importance of selecting an appropriate chassis for your system. And we talked about thinking about the characteristics your chassis would need to have in order to live, remember they're living things, and function in the environment that you have chosen your system to perform in. So we gave the example last time of developing a system that would be deployed out in the ocean. So we talked about how our chassis would need to be able to survive in the ocean, right? It would have to be salt tolerant. It would have to be able to tolerate a certain pH, certain light levels. So these are all important characteristics to think about. From there, you're going to define your inputs and outputs of your system. Your input is going to be the signal that triggers your system to perform its function. Okay, so that could be a few different things. It could be an inducing agent that you are going to provide for your system. So you're going to tell it when to do its function by providing the input. It could be a signal that's location dependent. Maybe you don't want your system to perform its function unless it's in a specific environment or a specific part of the body. So you would have to pick some sort of signal that would be specific to that location. Or the input could just be a compound or a molecule out in the environment that you want it to respond to. Then you need to define your output, which is the thing that you will be measuring as the indicator that your system is working. And it's important to stress here that your output is something that you can measure. Color is a very popular one. You do not have to use color or fluorescence, but those are very popular choices. Okay, so that's system level design. Then we moved on to device level design. And this is where you need to start defining the device or devices that will perform the functions needed for your system to work the way that you just defined. So again, you can be very vague at this stage. You don't actually need to know how to code the device to do that thing yet. We're going to get into that a little bit more today. So you can simply define it as whatever its function is going to be. If you want to have a color signal output, you can just say, I need a color generating device, right? Just name it for whatever the function is going to be. Okay. So we did talk last time about the possibility of needing more than one device to get your chassis to perform the desired functions. Remember that unless you choose to use a cell-free system, you will be coding living cells to perform these functions, right? So it's a little tricky. The device level design is where you can start to lean on your mentor a little bit more for help. They will definitely help you out with that. So looking at our example from last webinar, we set up this example where we wanted to design a cell that would detect arsenic in water and produce a color signal when levels are unsafe. So here we define the input as arsenic. That's what's going to trigger our system to function. And if the arsenic is present, the output would be a color signal. So to do this, we have to have more than one device, possibly. One device would be an arsenic detecting device so that we know if arsenic is there or not. And then to get our output, we need a color generating device. Okay, so for the next few slides, and actually the remainder of today's webinar, we're going to focus on this particular example. We're going to walk through how this would work. Okay, so the next few slides will be a little bit repetitive. The purpose in that is to show you a few different ways to think through and visualize how this system could work as we transition from device level design to parts level design. Because we all kind of think about these things differently. So I want to show you different ways of approaching it. Okay, so remember again, your devices will all be in the same living cell. So they will interact with each other. So to help visualize what devices and eventually parts you might need, it's important for you to think about how you want your system to respond under different input conditions. Okay, so here in our example, we want a cell that detects arsenic and produces a color signal when arsenic levels are unsafe. Okay, so that's our kind of example here. It's a biosensor. This is a very basic example where you're detecting one input and you're measuring one output. Okay, so we're going to look at this two different ways. On the left, I'm going to just have kind of like a visual, and on the right, we have what's known as a truth table. Truth tables are not my preferred method. I like the visuals, but we're doing the truth table in case that's more your jam. Okay, so in this example, if arsenic levels are very, very low, below a certain threshold, or they're zero, would you expect your system to produce any color? This is where you can throw some stuff in the chat. This is a simple yes or no. So in this system that we're envisioning, if we take a sample of water and there's no arsenic, would we expect our system to produce a color? Yes or no? What do we think? All right, I'm seeing some no. All right, yeah, so we would not want it to produce color if the arsenic levels are low. Good. So in our truth table, if there's no arsenic, no input, we would put a 0 or a minus sign. Okay, so in this example, if there's no arsenic, there's no color. So for output, we're also going to put a 0 or a minus, whichever way you want to do it. Okay. All right, let's look at the other response for our system. Let's say we take a sample of water and there is presence of arsenic, high levels of arsenic. Would we theoretically expect our system to produce a color? All right, good. I'm seeing some yeses. Yeah, so in this case, we would hopefully see color. That's how we envision this system working. So in our truth table here, for input, we would put a 1 or a plus. And for output, we would have a 1 or a plus. Okay, now we're going to get into this a little bit later, but I wanted to bring it up a little bit here. You can always come back to this stage of planning and development as you dive deeper into your design and you kind of want to fine tune your system or make it more complex. So for example, if you are designing this arsenic biosensor, you might later think, it might be helpful if we were able to detect different levels of arsenic. Maybe you want to set a threshold where if it's below this amount, that's okay. If it's between these two, you need to watch out. Or if it's above this, that's very, very high. Definitely don't drink, right? Or you might want your sensor to be able to detect another common water contaminant that people might want to watch out for. Okay, these are all possibilities. In those cases, you would revisit this stage and you would just simply add another column to your inputs on your table and think through how your system should respond if there's one of those inputs. What if both of those inputs are there? How would you expect it to behave? Okay. Same with outputs. If you wanted to be able to detect different levels of something, you would just add another output column and think through how you would expect it to behave. Okay. Okay, so how do we use this information now to visualize the system as genetic devices? All right, so here's our system. We've got our input, our output, our arsenic detecting device, our color generating device. So our first device would serve to detect whether or not the input, in this case arsenic, is present. So if arsenic is present, then that would signal our second device to produce color. Okay, great. That's our truth table where it was 1 for input, 1 for output. Now, under normal conditions or clean water conditions, we would expect no arsenic hopefully, or very, very low levels of arsenic, and no color. This would mean that we need the color generating device to be turned off if there's no arsenic. Okay. So if you want your output generating device to be turned on in the presence of your input, you need your other device to be turning off that output. Okay. I'm going to say that a few times in a few different ways because that's a little confusing. So in synthetic biology, we use that little symbol, the line like this, like a sideways T, to represent stop or inhibit. So we need a device that will turn off our color generating device until that input is needed. Does anybody know what fancy science word we use for turning off or inhibiting? It starts with an R. Give people a second to think here. We need a repressor, okay? So we need our first device to produce a repressor that will stop our output device from functioning unless that input is there. Okay, I'm going to show you how this works a few different ways because it gets a little tricky. Okay, so we know how we want our system to operate. So we need to start thinking about how the devices will function to carry out these tasks. And that's where we kind of transition from device level design into parts level design. Okay. So here we have on the top, we have no input. There's no input, no arsenic. That means that our arsenic detecting device is producing a repressor that's going to inhibit the color generating device. Okay, so it's going to block expression of that color generating device. Now, if there is arsenic present, now we have our input. That arsenic is going to interact with the repressor and remove it so that now that color generating device can function and we can get our color. Okay. Does that make sense to people before we move on? Give me some thumbs ups or yeses. All right, I see some yeses. Okay. Does it utilize a positive feedback loop? So it depends on the system that you're generating. Yeah, so in this case, we want it off unless the input is there. If the input is there, then our output will be on. When the input goes away, the output will be off. Does that help? I don't really think of that as like a feedback loop necessarily, but maybe it is.
Yeah, so I wouldn't I wouldn't say that in this case it's a positive feedback loop, no.
Okay. 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 So we can move on. All right. So now we're going to transition into parts level design. Okay. So this is where you're really getting into the sciencey part of this. So you're really thinking about what genetic components you need to make this device work, or make your devices work so that your whole system works. Okay. So there are two main steps to this. The first step is to kind of map out your genetic circuits and how those would work. And when we map out our genetic circuits, we call them circuits because it pretty much looks like circuit maps or circuit board maps if you're familiar with doing those. So there are many ways to do this part, so keep your options open. This can be a very frustrating part for a lot of people, myself included. This part can take a lot of time, lots of changing things around, so be flexible. So once you have your diagrams, then you can move on to step 2 and actually begin searching for the parts to actually build your genetic circuit. When I say actually build your genetic circuit, I mean like the DNA pieces to actually put together a plasmid that you would then put into your chassis. Okay, but first, before we can do this, we need to understand what these parts are. We keep saying parts, so we're going to talk about what these parts are. and we're going to talk about the language used to communicate genetic circuits. Okay, so we just spent a few slides talking through how we envision our arsenic biosensor system functioning. And as you probably noticed, it can be kind of hard to verbally describe that, and it can be a little hard to follow along these visuals, despite all the animations and stuff. So luckily, synthetic biologists have developed a universal language to help people communicate how genetic circuits work. That language is called SBOL, or Synthetic Biology Open Language. So we're going to do a quick overview of SBOL and function of the basic parts that you will be using. Then we'll use that synthetic biology language to make a diagram of our arsenic biosensor circuit. Okay, so synthetic biology open language. I gave you the website here. I highly recommend checking it out. It's a great website. It lists all the different parts and the universal symbols used. So these symbols are universally recognized. The colors here are arbitrary. I just kind of chose colors. You can choose your own colors. The parts that I have listed here are the very basic parts needed for a functional genetic circuit. There are lots more parts that you could use. They get more complex, like aptamers and RNA hairpins and loop structures and all that stuff. So these are really the basic ones. So these are the ones that we're going to focus on today. We have the promoter, the operator, ribosome binding site, or RBS. The green arrow one you might see defined a few different ways. It might be listed as an open reading frame or ORF. I think iGEM refers to it as coding sequence or CDS. There's some other, you might see it as like reporter. There are a few different things there, but you're probably going to see it listed as ORF or CDS. And then you have your terminator. Okay, so the promoter and the operator, oh, sorry, CDS stands for coding sequence. So your promoter is going to regulate transcription along with the operator. The ribosome binding site is going to regulate translation. And the terminator is going to tell you when you've reached the end of your coding sequence. Okay. Okay, so the operator is where any repressor that might be present in your system is going to bind. And anytime you're inhibiting or stopping a certain process, you're going to use that kind of sideways T symbol at the bottom. And anytime you're producing something or there's a process that leads to something, you're going to use an arrow. Typically in your circuit diagrams, you don't visualize the operator or the terminator. It's just understood that they're there. But for these beginning steps and learning stages, it's good practice to include them. Another thing about the terminator is terminators can be very leaky, meaning that some of the mRNA transcripts will have some excess code at the end. So to stop that from happening, it is fairly standard practice to use a double terminator. Just throwing that out there in case you happen to see that. Okay. Let's do a really quick overview of transcription and translation so that we understand how all of these parts function. So here we have just a basic gene, right? We've got our promoter, ribosome binding site, open reading frame or coding sequence, and our terminator. The promoter is going to be at the front. That's the element that kind of indicates this is where the instructions start to make my protein. So it functions by recruiting the RNA polymerase molecule to the gene of interest. So it'll attach at the promoter sequence. It'll read that DNA and produce mRNA. Okay. Now, copied onto the mRNA is going to be your ribosome binding site and your open reading frame or coding sequence, which will code for your protein product. Okay. If you want to regulate transcription, meaning how much mRNA gets made, which can affect how much protein gets made, you have a few different ways that you can do that. You can use a repressor system to turn transcription off. or on. You can control that. We're going to do that today. Another option is you can make the promoter sequence stronger or weaker. And when I say stronger or weaker, you can think of it as easier or not as easy for the RNA polymerase to find it and attach and stay attached. Or you can use two different types of promoters. There are two main types. There's a oh my gosh, I always say this wrong. Constitutive promoter, which means it is always on. It's just always available for RNA polymerase to attach and read and transcribe. Or you can have an inducible promoter, which means that transcription can only occur under certain conditions, usually only if a certain molecule or signal is present. We got a question about how do you make the promoter stronger or weaker? It has to do with the consensus sequence for the promoter or whatever sequence you're using. Are we familiar with consensus sequence? Consensus sequence is like the, it'd be like if you took a survey of your whole class and said, we're ordering pizza, we can only get one kind, what kind should we get? And the overall consensus is we want cheese, right? So it's kind of like that where you look at the sequence in the promoter, whatever the promoter is that you want to use, and you're going to look at that promoter sequence in a whole bunch of bacteria or whatever the host is, and you're going to look at the most common sequence across the board. So if you want to make it stronger, you want your promoter to be the closest match to that consensus sequence. If you want to make it weaker, you want to kind of jumble up the letters a little bit, the nucleotide sequence a little bit, so that it doesn't really match the consensus. Okay, all right, I hope that made sense. Okay, so that's your transcription. So we've made our mRNA. It has our ribosome binding site that serves to recruit the ribosome so that it can translate it and make our protein. Okay. So similar to the promoter, we can change the strength of our ribosome binding site to be easier or more challenging for the ribosome to latch onto and translate. And that's kind of the basis of our iTunes device lab. If any of you watching have come to the Learning Lab for a workshop or you were part of our summer research program, you've probably done the iTunes lab. It's in the BioBuilder book. And that is a lab where we have adjusted the promoter and ribosome binding site strength in E. coli, and we look at how that affects the production of beta-gal protein. Okay. All right, so now we're going to look at combining genetic circuits to regulate gene expression. So we're going to use an example of a biosensor, kind of like the arsenic one, but we're going to be more general. So a biosensor that fluoresces green when a certain chemical is present. Okay, so that's going to be our example. All right, so here we just have those basic parts in that order, right? Promoter, operator, RBS, open reading frame. So here, I'm going to start backwards. I like to start with my output because to me that makes more sense. So I'm going to show you this example starting with the output circuit and going backwards. And then our next example, we're going to start with the input and go forward. Okay, so we're going to start with our output circuit. So we want a biosensor that fluoresces green as the output. So we know that we need a circuit that codes for a green fluorescent protein, GFP. All right, so we have a circuit that will produce that. We have a promoter that will recruit RNA polymerase. We've got ribosome binding site. We've got GFP. Great. Now we want to build a biosensor that only fluoresces green when a certain chemical is present. So you tell me, do we want this circuit, this GFP circuit, to always be on? Yes or no? What do we think? Good, good. All right, good. I'm seeing some no's. Excellent. Yeah, we don't want it to always be on. We only want it to be on when the chemical that we're looking for is present. Okay, So what will we need in our system to make sure that this circuit is not constantly on and making GFP? What will we need? Hint, I mentioned it earlier in our arsenic biosensor. Do we remember? It starts with an R. Yes, yes, I'm seeing it. Good. Yeah, we're going to need a repressor. So we're going to need a repressor molecule. And repressor molecules function by binding to that operator region just in front of the promoter. Okay. So here we have a repressor molecule. So it's going to function by blocking RNA polymerase. The RNA polymerase can still kind of bind to that promoter region, but it can't, it's got a roadblock. It's got something blocking it. It can't move forward. All right. So the repressor being bound to the operator is going to prevent transcription. So we're not going to get protein expression, no GFP, no color. Perfect. All right, we've got that part situated. But where does this repressor molecule come from? So a repressor molecule that specifically binds to an operator that we synthetically produced, it's not going to just magically be there in the cell, right? So where does it come from? Well, we have to build a genetic circuit to produce that repressor in our system. Okay, so we're working backwards. We need a repressor. Oh, we'll get to that. So someone asked, how does the repressor detach? We're going to get to that. Okay, so we have to code for the repressor so that it's there. So we're just going to do that by making another circuit, a separate promoter that's attached to a separate coding sequence that codes for our repressor protein. Okay. Okay. You were like one slide ahead of me. But how do we remove the repressor to turn on protein expression? So we don't want to block the GFP forever. We want to remove that repressor when our molecule is present, right? So we have these two genetic circuits right now, one that produces GFP and another that turns the GFP expression off. Okay, so how do we finally get the GFP production? Our input molecule can bind to the repressor. This is a very common way of doing it. This is the simple way of doing it. So you would choose a repressor protein that's able to recognize and interact with your molecule of interest. That would be your input or your inducer. Okay, so when those two bind, it changes the shape of that repressor, and now it can't bind anymore. So that's going to free up that section of DNA, and now the RNA polymerase can go ahead and do its thing, and you're going to get your GFP product. Okay? All right, so we just successfully mapped out the genetic circuits needed to produce that system. But as you can see, that took several animated slides and a whole bunch of mumbo jumbo from my mouth to explain it to you. We want to simplify it into just one visual diagram using the synthetic biology open language. Oh, we had a question. All right. I'll remember to get to that. Chloe, remind me about that question if I don't get to it. Okay. So here's what it would look like if we turned all those slides into just one diagram. And this is what you will visually present, ideally at the final assembly for your system, something that looks like this. All right. So we have our circuit with the promoter for the GFP. Sorry, mine says RFP. I forgot to change that. That's coding for the GFP output. But we also have our promoter regulating our repressor protein. And we're showing that it's a repressor protein by putting that little T, the inhibiting symbol, next to the GFP promoter. And then we show that our inducer molecule, which is the I here, would inhibit our repressor by, again, having that inhibition symbol. Okay. If you're a truth table person, I have the truth table here. So just like before, without the input, so if you removed that purple inducer molecule, then you would have repression of the GFP, so no output. If the inducer molecule is there, it's going to inhibit the repressor, so you will get your GFP. Okay. Now, when you're putting yours together, there is an awesome free website called BioRender that I highly recommend you use. I have an image here of what its workspace looks like. BioRender already has all of the synthetic biology open language symbols built in. You literally just click on whichever ones you want, drag them into place to build your circuits. You can change the color to whatever you want. It has all the arrows and symbols. You can add text. So it's a super easy way to map out your diagram. So definitely check that out. The link that I have here for the video under BioRender is an amazing video that was put together by an iGEM team a few years ago that gives you an awesome tutorial of how to use BioRender for genetic circuits. So definitely check that out. Okay, so now that we have a little bit of experience with genetic circuits, let's do this for our arsenic biosensor for water. Okay, so we're going to practice with this system. So in working through this system example, we're also going to go that next step and also talk about how to actually find the specific parts that you will need for your circuits. So not just saying a coding sequence for color. We're going to talk about how to find that. Okay. All right. So here's our system again. Our input is arsenic. Our output is going to be a color signal. We haven't defined it beyond that yet. We're planning on 2 devices at this stage, one that will detect arsenic, but will also serve as a repressor for the color generating device unless our input is present. Okay. So we need 2 circuits then, one for each device. So we're just going to map them out real basic like this. So our first circuit will code for a repressor. So we know that that coding sequence in the first circuit will be a repressor. We don't know which one yet. And we know that it will inhibit the promoter of the second circuit. Okay, so those are some clues. If you guys like doing like murder mysteries and things like that, this is right up your alley. And we know that the second circuit will code for our color output. So our coding sequence in the second circuit will be some sort of color, right? And note that we also need our system to respond to arsenic. So that's an added layer there. And it needs to respond to it by turning off the repressor. All right, so this is what we got to sort out here. So we're going to take a closer look now at each circuit to determine what parts we need to make our system function this way. We're going to start with circuit 1 this time. Last example, I started at the output and worked backwards. This time we're going to start with circuit 1 and go forwards. And we're going to determine what we want to use for our coding sequence here, the repressor, and what we're going to use for our promoter. All right. So for coding sequence 1, we're looking for a protein that will inhibit transcription of circuit 2 and is responsive to arsenic. That sounds like a crazy random thing to try to find, right? Well, a quick Google search for just those 2 characteristics, a transcriptional repressor and a protein that is responsive to arsenic, if you just search those, you're going to identify what is known as the Rs-R protein. And that protein is part of the Rs operon. So the Rs operon is a very well-studied system that manages arsenic detoxification in bacteria, including E. coli, and a few other types of bacteria also have this. So it is well-studied. We know that Rs-R binds to the Rs promoter and represses transcription until arsenic binds to it, which is exactly what we're looking for, right? We're looking for a protein that will repress a promoter, but if arsenic binds to it, it will be removed, right? So perfect. And I totally admit that this is like a little bit of cooking show magic right now. And I'm just like, oh, presto, it took 2 seconds to find this. It might take you a really long time to find just the right repressor or just the right promoter for your system. So please don't get discouraged. You might not even find something that works for your system. And that's totally fine. Again, that's why we tell you to kind of be flexible as you move through this process so that you can kind of fall back on other ideas, take different routes. This is where you really lean on that mentor help. They are the experts at this and can kind of guide you here. Okay. All right. So we have our coding sequence for circuit one. We know we want the R's are repressor protein. All right, so now let's think about our promoter options for circuit one. So we need to decide what type of promoter to use to control expression of our repressor protein. So we need to think about when we want our repressor to be expressed. Do we want our repressor to be expressed all the time or only certain times? So remember, our R's are repressor, the repressor protein, that's our arsenic sensor. So it's going to turn on color expression when arsenic is present. So do we want our arsenic sensor to be there all the time or not? What do we think? Yes or no? Ooh, I'm seeing some mixed thoughts here. Yeah, so this is a little tricky. So we actually want it to be expressed all the time. So hear me out here. Hear me out here, people. So we want it to be we want it to be there all the time so that there's no color unless the arsenic is present. Okay. If our arsenic detector is not there all the time, we might miss the arsenic. So you want your detector to be there all the time. That'd be like if you have smoke detectors, right? But they get turned off certain times of the day. That wouldn't be very helpful, right? You want your smoke detectors turned on all the time. Okay, good. Okay, so in this case, we only want our system to produce color output if arsenic is present. If arsenic is not present, we want the color generating device off. So we want the first circuit to always be on and always producing that repressor protein. The repressor will be removed if the arsenic is present. Okay, so since we want it to be on all the time, remember we talked about two types of promoters. Would we want a constitutive promoter that's on all the time, or do we want an inducible promoter, which is only on certain times? Which one do we want? We want to make our repressor all the time. Keep it coming. Good, good. Yeah, so we want to use a constitutive promoter here so that transcription can just keep happening and we keep making that repressor protein. Good. Okay. Now, as you're building these systems, if you're a BioBuilder Club who's been around for a while and you've been working on this project for a while and you're actually like testing and building things, you might get to a point where you have to like fine tune, okay, how much repressor does our system need to produce? And that's where you would kind of start to fiddle with the promoter strength or the ribosome binding site strength. If you're not there, that shouldn't even cross your mind at this stage. So don't worry about it. All right, so for circuit one, we want a constitutive promoter. Okay. Okay, so let's go back to this. So we have figured out our first circuit. We want to use a constitutive promoter, and it's going to control R's R. That's going to give us our repressor that controls the second promoter, and arsenic can bind it and remove it. Perfect. All right, now we're going to figure out circuit two. So we want circuit two to be repressed by ours are, and we want it to produce a color output. Okay, so we need to think about what we want to use for our color output sequence and what type of promoter we want to control that expression. All right, let's start with the easy one. Let's start with the coding sequence here, our color generating output. You can select so many colors. There are so many out there. So think about how you would utilize your device. If certain colors would be easier or harder to see depending on how you're using it. And once you select a color, you can search databases that I'll share with you in a few slides for coding sequences of naturally occurring pigments. You can always change this protein out as your idea develops. This is an easy one to kind of swap out if you're doing color. So let's say we chose red. There are tons of natural pigments that produce red cabbage, watermelon, carrots, all those. So there are plenty out there. All right, now the promoter to control our color generating device. The tricky part here is we need that promoter to recognize our repressor protein. Luckily, we already discovered our repressor protein binds to the R's promoter. So that's already a known thing. So that's the promoter we're going to use to control circuit 2. All right. So we're going to put all of this together into a genetic circuit here. And before we map out like our final professional looking one, we're gonna walk through and make sure this would function the way we want it to. Okay, so circuit 1, we've got our constitutive promoter. It's always transcribing and pumping out our R's are repressor protein. That protein recognizes and binds to the R's promoter. Great, that turns off our red color generator. Perfect. Okay. We also know that R's are, if arsenic is present, it will bind to it. If it binds to it, it's going to remove it from that promoter. That will allow transcription and translation to happen, and we should see our red color. Great. So this is just one example of how you could possibly design a genetic circuit for this system. Your design might have more circuits. It might have less circuits. Your team might use a repressor system. You might not. Okay, I'm just giving this example because this is a little more complex thinking. So I wanted you to understand that. You might want to detect various levels of a contaminant instead of just if it's present or not. So if you would want to do that, that's where your ribosome binding site would come into play. And you can adjust that to tweak how much of your protein you're producing. Maybe you want to tweak how much red color. So you could detect, is it a strong red color or a weak red color? That sort of thing. Okay. All right. So in this example, we used constitutive promoters to control our repressor. So let's quickly talk about when you might want to use an inducible promoter. So we have not done that yet. Okay. So an inducible promoter, again, is only able to turn on transcription under certain conditions. These are living cells, and it takes energy for them to produce proteins. You also don't want your cells wasting energy producing your protein unnecessarily. The protein product might be toxic to your host cell at low levels or moderate levels or high levels. So let's go over a few examples of when you would use that inducible promoter. One example might be when you want to have high yield production of a protein. So again, if you're dealing with biofuels or producing pharmaceuticals, a really easy way that you can boost production is by growing your culture first so that you have happy, healthy, fed cells. And then you tell them, okay, now make my protein. And then you'll get a lot of protein. Another time you might want to use an inducible promoter is if the protein product you're trying to make is harmful to your host. You would want expression of your protein to be off until you needed it to help reduce stress on your cells and cell death. So in these two situations, your system's input would be your inducing agent. So two really popular examples of inducing agents that get used in these types of situations. One is IPTG, and that comes from the lac operon system. The other is doxycycline, which is an antibiotic, and that comes from the TET on, TET off system. Okay, so if you wanted to use an inducible system like these, you would incorporate genetic circuits with the lac system or the TET system to use those in inducing agents. Okay, another time you would use inducible promoters, you may only want your system to be turned on when it's in the right environment or location. The classic example of this is drug delivery systems. So you might want like timed release, right? Or if you're detecting tumors, you don't want to inject someone and your protein medicine product is just like all over the body. That's not very effective, right? You want it to only pump out that once it's in the right location. Right. So in that case, you would use an inducible promoter that's responsive to an input that's specific to your desired location or environment, or an external stimuli. So for example, let's say you wanted to design a system that releases a protein to help with H. pylori infection. H. pylori is in the gut, in the stomach. So what might your system sense as its input, its trigger? What would be the environmental trigger that says, oh, I'm in the stomach? Can you think of something that would kind of trigger something or tell you whether you're in the stomach or not? Oh, good. I see it in the chat. Yes, pH. Excellent. So pH would be a great input in that example. So you would have, you would incorporate a circuit, a genetic circuit that could detect pH, where once it's in a specific range, that's kind of specific to the gut, that's when it would trigger release of the protein that you wanted to release. Okay. Can we think of any other types of stimuli, environmental stimuli that could be used as inducers for drug delivery? What do we think? We had pH. What else? Oh, someone said glucose. I didn't think of that. Maybe. Time. Time is excellent. Yep, you can do time. You could do, oh, no one's saying the one I always go to. Temperature is a big one. So temperature, you can use specific biological markers that might only be present in certain locations or parts of the body. You can also use electrical stimuli, which we'll talk about in a second. Yeah, someone said proteins. So proteins would be like biological markers. So in the example I have here, this is from a paper where they wanted to trigger encapsulation of the bacteria cells. The point here was they wanted to deliver drugs specifically to tumors. And if it's a bacterial system, as soon as they're injected into the body, in this case mice, the immune system would immediately attack it, right? So to do that, they would encapsulate the bacteria cells, and they did this by an inducible promoter. So they told the bacteria when to encapsulate, and then over time, when that induction went away, that inducing agent went away, the encapsulation turned off, and then they could be cleared from the body. All right, so that's a cool way of ensuring that they survive long enough to deliver it, but then they can actually get cleared out. Another really quick example, we're getting short on time, so I'm going to be quick about this one. This is one that we have our summer research program students read. I like this because it's a combination of SynBio and electronic devices. So this group wanted to monitor GI system health in real time. And they did that by making this little like biosensor that had the E. coli in it. And they did it where, and now you can all read genetic circuits, it produced a protein that responds to heme, which is part of blood. And when it responded to the blood, it turned off a repressor, and you got luciferase expression, luminescence. And their little electronic board would sense the luminescence and send an electronic signal to an external device, which was pretty cool. Okay, something we haven't gotten into yet, but is very, very important. So we're talking about modifying microbes, living organisms, typically bacteria or yeast. Your group might want to build a system that you don't want released into the environment long term, or don't want in people's bodies long term, because we don't know the impacts. So in this case, you would want to incorporate something called a genetic kill switch. And this is kind of like a safeguard biocontainment tool. It's going to prevent any unintended survival or buildup or spread into the environment. You wouldn't necessarily need a kill switch for something like a biosensor, like if you're using like a test strip type thing. This would be more if you're injecting it or ingesting it, right? The example here is shown from a 2020 iGEM team. They incorporated a toxin antitoxin circuit that was under a constitutive promoter. So what you're seeing here is the toxin is always being produced that would kill the host cell. The antitoxin is under translational control of a ribosome binding site that only functions over 37 degrees Celsius, which is body temp. Okay, so they only want it to function inside the body. Once the person theoretically poops it out, right, the temperature is going to change and it's going to drop significantly. And that's going to inhibit the action of, or sorry, the translation of the antitoxin. And now you're only producing the toxin and it's going to self-destruct, which is pretty cool. All right, really quickly, how do we go about actually finding these parts? I did, again, cooking show magic and just like told you parts that exist. You actually have to find them. So we're going to give you a few places to do that. One is the iGEM parts registry. They actually have two right now. It's a little complicated. So they're undergoing construction of a new and improved parts registry. It's a little finicky. I don't know. I was having some trouble with it. So the old one is still there. You can get to it using this link here. You're going to click on catalog and you're able to explore parts by type, collection, frequency of use. So for example, you would see a list like this. So you can search for promoters, ribosome binding sites, protein domains, protein coding sequences, all that stuff. Let's say we want to look at promoters. Here are your options. You can click on constitutive promoters. You can look for metal sensitive promoters, like for our arsenic one. It's all there, okay? Protein coding sequences. If you click on that one of the options is transcriptional regulators, like repressors, right? So you can actually search just for repressors and it'll give you a list, okay? Another place to look is the iGEM distribution kit plate database. The link is here. I gave you a shortened link for it. This one is more like a spreadsheet. It's nice and pretty and colorful. And you can sort it however you want. You can sort it by fluorescence or by just coding sequences or promoters or whatever it is you're looking for. Okay. So if a basic circuit diagram for your system is as far as you get for the final assembly, awesome. That's fine. If you have time to spare, or you're a more established BioBuilder club that already had a project in the works, now you could probably start moving on to actually building your genetic system by finding the DNA sequences of your parts and starting to build your plasmids and transforming your chassis and testing your system. But again, we totally understand the research involved in getting just a circuit and parts nailed down. So if that's where you're at, that is great.
So we do have a plasma design webinar from December, so I'll make sure that you get that link too, because that was really well with this presentation.
Oh, we had a question. That Chloe, that might be a better question for you. I don't know if you saw it.
Um, in the meantime, while I read that question, do you want to answer the previous question about
Oh, yeah,
if
where
the
was that?
repressor can't be removed?
Oh,
Here,
yes.
I
Let me
copied
scroll
it.
back to that
Let me
one.
get there.
Ba, 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 ba. Okay, there it is.
I
What happens when the
recopied it.
repressor can't be removed by the chemical being sensed? So, yes, you might encounter a situation where you found a repressor that works for your system, but that repressor does not respond to the input that you have in mind. Um, that happens sometimes. In that case, you will need some sort of signaling pathway where your input is sensed by something else that would then interact with the repressor. I know that's not a very specific response because there is no specific answer. You just have to kind of find a way to connect those two things. Okay. Does that make sense? Did I answer that well enough? Yes, it is very unpredictable. Yeah, and you'll see that once you start doing research and looking at papers, you'll see that sometimes the genetic circuit looks all crazy because they had the input had to interact with something else that could be recognized by the repressor. Yeah, okay. All right, any other questions? Let me see. All right. Can we see, and I can show you, let me see. I think you can see the my screen. Can we see this? So this is the synthetic biology open language website. Yeah. Okay, good. So this is the iGEM parts registry website. You can see all the different categories of proteins. You can find transport proteins. You can do receptor proteins, proteases. This is what the new iGEM parts registry looks like. I don't like it as much yet. I don't know. Hopefully it gets better. But I'll show you an example. So you can type in like you can type in here like kill switch and it should search. Yep. So it gave me 217 results and there's all sorts of different kill switches here I can choose from. Okay. We can search for arsenic. Um, there's our R protein, right? So it gives us a bunch of arsenic sensing things. And on the left-hand side, you can choose like what part type you're looking for. You can see there are lots of part types. We only got into the very basic ones. Um, so yeah, have fun with it. All right. People are excited. Good. All right. Any other questions?
And if you think of questions later, feel free to email me and I can answer specific questions and also send resources. And if your team is working with a mentor, they are a great person to ask, as Danielle said, specifically when you're looking into, I get back 132 results. Which one should I select? Any last questions?
Hey, I just wanted to say thank you. I definitely will save this video for future references. Thank you.
Yes, and the plasmid design webinar is from Anna White. So I think these two go together really well.
It does. So, thank you.
Well, thank you, Danielle. This was very, very helpful. I appreciate it, and I will make sure everyone gets access to this resource.
Great.
Thanks, everyone.